Hello students, today we are going to observe different stages of mitosis and meiosis under microscope. You have already studied this chapter and you have cleared your concepts about the mitosis and meiosis. Today we will go to the theoretical part, then we will move to the lab and we will observe under the microscope. We will use different slides of mitosis and meiosis. We have already the permanent slides. So one by one we will try to focus on the particular section so that we have a keen observation over there. Let's start with this. We have the mitosis and the meiosis. Mitosis, you know very well. It will produce two daughter cells which have the same ploidy. It means there is no reduction in the chromosome, whereas meiosis will result in production of four daughter cells. If there is production of four daughter cells, it means that the chromosome number get reduced or the ploidy get reduced. Some features mitosis. We have 5 stages including our interphase. Same in meiosis, we have 2 stages, meiosis 1 and meiosis 2. We have 10 stages in total. And interphase. Mitosis, it occurs in somatic cells where it occurs in germ cells. There are few common things among these is same what both produce new cells. They have all the basic steps and they all start with the single cell. In the meiosis, we particularly we study that it helps or it forms the germ cells or the gamete cells. We have different phases of mitosis and meiosis. See, prophase, metaphase, anaphase, telophase, and then followed by cytokinesis. Before formation of two daughter cell, every cell, it prepares itself to make 2 copies. So these steps include all these preparations before becoming 2 cells in mitosis. These stages manage condensation, alignment, separation and segregation of chromosomes, and after that it will go for cytokinesis. For your reference, we have placed some clear pictures to understand the concept of the mitosis. These pictures, these diagrams, these are taken through the microscope, electron microscope, not our common microscope, what we are using in our lab. And this is the keen observation. If we see the prophase, this one and this one. Okay, this one is the keen observation or the aligned observation. This is the real picture where the chromosome become distinct. Another one is metaphase. There is an equatorial plate where chromosomes align themselves. In anaphase, this one, definitely the chromosomes are moving towards the pole. This one. After that, the telophase, and there is completely division of the cell. It means that the cell produces 2 daughter cells. This is the concept of mitosis. Whereas in meiosis, we have meiosis 1 and meiosis 2. One is called reductional division. Why reductional division? In meiosis 1, the chromosome reduced to half, but in meiosis 2, the chromosome remain same. So this particular step mimic of the mitosis or resemblance, it will give the resemblance of mitosis. We have 10 different stages including the prophase 1, metaphase 1, anaphase 1, telophase 1, prophase 2, metaphase 2, anaphase 2, telophase 2, and after that we will go for the cytokinesis and it will give us the 4 daughter cells which has the half ploidy or reduced half chromosome numbers. Now what we will do now, we will take our slides one by one under the microscope and we will try to shoot it with the help of camera so that you can get a real picture. When we will observe under microscope, step by step I am going to explain you the main observations or the main steps. Let's move to the lab. Dear students, we have different slides. We have slides of mitosis and meiosis, which we are going to observe under microscope. Okay, now before that we have to consider few precautions. Handle these slides with care, as they are the permanent slides and they may get damaged if used roughly. Wear gloves so that they can protect us. Now, one by one, we are going to start to observe this under microscope. First, we will go for the mitosis. Let's start with first slide, that is the mitosis and the prophase. Okay, students, this is the prophase of mitosis, where chromatin condenses into visible chromosomes. Centromere moves opposite sides of the cell, forming the mitotic spindle. Okay. Now here there is no clearly visible nucleus. Nuclear membrane also begins to disappear. Students, this is metaphase, chromosome aligned at the equator of the cell. Metaphase plate formation, we can see it clearly. Spindle fibers, they are pulling the sister chromatids with equal forces. So, accurate division may occur. This slide is of anaphase, sister chromatids separated, centromere pulled towards the poles. Okay, I hope it is little bit more clear or visible. My dear students, this is telophase. Chromosome reaches opposite poles and begins to condense. Nuclear envelope slowly reforming around the two sets of the chromosomes. Okay, students, I hope you are getting here the two cells. This step is of the cytokinesis. Okay, cytoplasma divides, forming two distinct daughter cells. Dear students, we have seen the slides of the mitosis. Now we will proceed to next step, that is the meiosis. One by one we will see all the steps. Okay, my dear friends, this is prophase 1. Chromosomes condense, homologous pair align. इसको हम we call it synapses. Crossing over occurs. Okay, this is the prophase 1. Dear students, this picture is little bit blur, but this is the metaphase 1. Homologous pair align along the cell center. It is forming tetrads. Dear students, anaphase 1, homologous chromosomes pull apart to opposite poles, sister chromatids remains attached. Telophase 1, we also getting few hints of cytokinesis here. Chromosomes reaches the pole and cell divides into two haploid daughter cells. Now we will see the meiosis 2. Two chromosome condense again into two new cells. Students, this is metaphase 2. Chromosome aligned at the equator in the both cells. Two sister chromatids finally separated and move towards the opposite pole. Telophase and the cytokinesis, nuclear membrane reforms around the 4 distinct nuclei and cell divides, resulting in 4 haploid gametes. Students, I know it is little bit difficult to observe under microscope, but with the help of teacher, with the help of keen observation, you will able to understand these steps. So thank you. Have a nice day.